Good evening, everyone. Welcome to the Kaapi with Curiosity talk. I request our director, Dr. B.R. Guruprasad sir, to welcome you all formally and also introduce today's speaker.
Our respected speaker of today, Professor Shweta Ramdas, and my colleagues from JNP, uh, ladies and gentlemen. Uh, Indeed, I'm honored to welcome you all to this latest lecture under the series Copy with Curiosity, jointly hosted by ICTS and JNP. So, on behalf of both these institutions, I welcome you all. As I told you, once again, let me say, I'm honored indeed to introduce today's speaker, Professor Shweta Ramdas, who is speaking on a very interesting subject, which is there already. What's in your DNA? Tales from the Human Genome. I didn't want to make a mistake, that's why I read it. So, all said and done, I'm sure it will be very lively and you will enjoy it. And as I was discussing with the uh, professor today, so the word DNA has entered everyday lexicon, in fact. Okay, DNA, genetic engineering, genome, all these things we are all very familiar, even as excuse me, even as lay people. Okay, the applications are numerous. So, what DNA and the related research can tell us about our species, the human evolution, and other possibilities, in fact. So, that's what the speaker is going to deal with in her talk today. And one more important thing is nowadays, this study of genetics is becoming much more important because of its importance in finding out the predisposition of certain humans, okay, individuals towards certain diseases, okay. So, and in addition to that, in forensics and such things, it finds wide application. So, I don't want to preempt all these things, but anyway, let me introduce the speaker, Dr. Shweta Ramdas, who is an assistant professor at Center for Brain Research in the Indian Institute of Science campus. And she earned her Ph.D. in Bioinformatics and an MA in Applied Statistics in 2018 from the University of Michigan. Professor Ramdas did her postdoctoral work at the University of Pennsylvania, where her work focused on identifying genetic variation in human diseases. Before coming to Center for Brain Research, Dr. Ramdas was an assistant professor at Azim Premji University from December 2021 to January 2023. Professor Shweta Ramdas research is at the intersection of genetics and data science. It's motivated by exploring the genetic variation in human populations in the context of human disease. That's what I was mentioning to earlier. Her group at CBR primarily uses bioinformatic and statistical approaches to address these questions. Once again, I welcome you, ma'am, for your kind gesture to come here and give a talk under the series Copy with Curiosity. So both ICTS, on behalf of ICTS and JNP, I thank you and I welcome you before that. Thanks a lot. Please. And
So there is a memento on behalf of ICTS. I request our director to please hand it over to the speaker.
Thank you. Thank you. Start. Can everyone hear me okay? Okay, good. Hello, everyone. Thank you to the organizers for the opportunity to speak here. Speaking at the Planetarium is really a dream come true. So I'm more excited to be here than anyone else in this room, I think. Um, Over the next 45, 50 minutes or so, what I was hoping to do is give you a little bit of a glimpse into the world of human genetics. I'm a geneticist, and I've been sort of fascinated with human genetics and DNA for the last 10, 15 years. And I'm sure everyone in this room is also shares that fascination because it's such an integral part of what makes us us. So I'm really hoping to convince you that this is something like beautiful and exciting, and hopefully we can have a conversation about that. So before I go into the story, I thought we'll first get on the same page and go a little bit of biology. So what is your DNA, or what is your genetic data? Each of us carries millions of cells. So if you look at your own body, you have millions, perhaps billions of cells, depending on how you count it. And now most of these cells carry a copy of your DNA, which we'll call the genome. And this genome is very, 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 very long. So each cell contains your genome, which is 3 billion letters long. If you want to get a sense for what 3 billion is, it's like 500 copies of your favorite novel, right? It's huge. It's so large that it's actually difficult to really grasp how large your genome is. If you look under the microscope, you see your genome arrayed out into these things called chromosomes, where you have 23 pairs of chromosomes, one from mom, one from dad, right? So each of our cells has your genome arrayed out into these chromosomes. And within these chromosomes, when we think of DNA, we think of genes. The analogy I'd like to use is that a gene is like a paragraph in a chapter. So if your genome is a book, your chromosome is a chapter. Let's say each of us carries 26, 23 times 2 pairs of chapters. A gene might be a paragraph in a chapter containing a message, and the message that your gene carries is a protein, right? Your gene contains the code that tells your cell, make this protein at this time in my cell. And another word we think of when we think of DNA is mutation, and I think of them as typos in the book, right? If you have mistakes or differences between one copy of, let's say, your Harry Potter book versus somebody else's copy of their Harry Potter, right? That's a typo, and in the context of DNA, that would be a mutation. And all of this is being carried out in the language of DNA, which has 4 letters, A, C, T, and G, instead of the 26 that we think about for English. So that's sort of what your genetic data is. I know the Kaapi series has a lot of astrophysics lectures, and there's a huge interest in stargazing, and that's a wondrous kind of science that all of us love. I kind of think of genetics and of my job as sequence gazing, where a lot of what we do is staring at these billions of letters in the genomes from hundreds of people and really trying to find the patterns that govern our lives and biology. So coming back to the point that was made earlier, which is why is genetics everywhere right now? And I will use the analogy of another field that is everywhere, AI, right? And why is AI everywhere right now? This graph kind of talks about that, right? If the reason AI is taking over our lives is because the cost of computing has become really low over time. So if you look at from 2001 to 2022, for the same kind of compute that you want to do, the cost has come down drastically, right? From like a millions of dollars to, let's say, $100,000 for whatever task you want to do. So let's compare, and so the availability of computing become easier and easier and easier has allowed us to deploy these AI models with greater and greater and greater ease. So how does that apply to genomes? If I look at the cost of the human genome over time, overlaid on that same graph, now the cost of genome sequencing has actually gone down quicker than the cost of compute. And this is kind of the main technological advancement that has made genetics such a big field right now, where it's becoming a lot easier to sequence DNA. It's becoming very easy to sequence thousands of genomes, even millions of genomes. And so now we have all of this data available, this genetic data, that allows us to really ask interesting questions that could not be asked maybe even 20 years ago. So the field of human genomics is really driven by this technological advancement in engineering, really, which has made sequencing so much more affordable. So the genome is, as I said, is huge. If I printed out my genome as physical books and arrayed them on a shelf, this is the amount of space it would take. And this genome contains thousands and perhaps more, millions of interesting stories, and I can't go over all of them in 45 minutes. So what I thought I would do is pick a few anecdotes from this library and share some of the stories that I think are the most interesting about the human genome with you. So the first story I want to talk about is about this one gene called LCT. And you might guess what LCT stands for. It's actually a gene called lactase. And most of us, milk is an important part of all of our lives. And but we also hear of terms lactose intolerance, lactose persistence. So some of us find it difficult to drink milk when we're adults, right? And we often have digestive problems, and that's called lactose intolerance. That's because our bodies don't produce an enzyme called lactase, leading to a buildup of the sugar called lactose that's found in milk, in our intestines, in our stomachs. Bacteria are then sort of have to consume this lactose, and the byproducts lead to bloating, cramps, all of the symptoms that one associates with lactose intolerance. On the other hand, those of us who are luckier and can drink as much milk and all of the mithai associated with milk without any problems, our bodies produce this enzyme called lactase. Usually this enzyme stops getting produced once you're an adult, but our bodies keep producing these, which means the lactose is broken down easily, we have no problems, and life is good. So what is not actually obvious is that we are really weird. Humans are weird because none of the other species that produce milk have lactase persistence. Pretty much every species where the calf or the pup drinks milk loses the ability to consume milk into adulthood. So humans are a huge outlier. There are no other examples of this happening. So how did this happen? How is lactase persistence a thing in our species? In fact, if you look around the world, this is a plot showing you the frequency of lactase persistence. You see that quite a few areas in the world have high levels of intolerance, right? Like, for example, Southeast Asia, some parts of Africa, whereas other parts have a high degree of lactose persistence, like Northern Europe, some parts of Northwest Africa, the Middle East, and so on. So there is a variation in our species' ability to digest lactose in adulthood. How did that come about? The answer lies in our genes, and I'll talk a little bit about that. So a bunch of researchers in 2002 were very interested in asking, why do some humans have lactose persistence? And geneticists really like families. We love studying families because we can collect these people in a family, separate them into categories, people with a certain trait, people who don't have a certain trait, and look for genetic differences between the two, right? So they study these 9 families and mark down which people in these families had lactase persistence and which people didn't. So in black, you see the people who could not digest lactose in adulthood, and in white are the people who could, and then find which parts of the genome differ between these two groups. And what was the hypothesis? You might assume that there's something to do with this gene that actually produces that lactase enzyme, right? So you might think that before the study, you might have thought that individuals who could produce lactose in adulthood had a different version of this gene. But what did they find? They actually found this one position that was a little away from this gene, about 14,000 positions up, we call it upstream of this lactase gene, that differentiated people who could digest lactase into adulthood, so lactase persistent people versus lactose intolerant people. So having a T allows you to produce lactase into adulthood. Why is that? How how could that variant, which is a little far away from that gene, what could that be doing? They found that having a T at this position actually influences whether this other protein called OCT1 can come and bind to this piece of DNA, right? So having a T at this position means this other protein comes and binds to this piece of DNA. And the DNA then does this cool looping thing in the cell where this part of the DNA loops with this lactase gene. They come close together. And the presence of this OCT1 protein tells the cell, listen, switch this on. I want this lactase gene to remain turned on. Do not turn it off. If in the absence of this, it's less likely that the lactase gene would be turned on. So that's what happens. Individuals who have the T have this cool sort of switching, the circuitry taking place in the cell throughout life, and they have lactase persistence. And individuals with the C, all of this is not happening. So the lactase gene is more likely to be turned off in adulthood. So this is, the mechanism they found. The researchers went in and actually put in this T into rats and found that when you put this piece of DNA with the T into a rat can now, start digesting lactose even into adulthood. So this actually kind of goes from finding a DNA sequence to actually seeing it in a live organism. But then other researchers said, wait, listen, it's not only in Finland that you see lactase persistence. What about all those other regions of the world? So they went and looked for the same thing in African families. There, in African families, you might have thought, okay, it's again that same position, right? That same C to T change that leads to lactase persistence. But there they found two other variants in that same nearby region, all that again confer the same property, where having a G to a C at that first position or a G to a C at that second position, again, are different ways that the cell uses to keep this lactase gene on in adulthood. So essentially what they saw is that in Northern Europeans and in African populations, there has been what's called convergent evolution, which means that independently, our genomes have evolved in response to this selection for lactase persistence, because it is so advantageous for humankind. And just to talk about what that might look like, right? I've spoken about everything from a sequence level, but from a population level, what might that look like? So let's say there was a group of people living somewhere, maybe in the Middle East, in Central Southern Europe, around 10,000 years ago. Most people had that C variant, right? They could not, our default state is that we couldn't digest lactose into adulthood. So most people had that C mutation variant, but a few people, very few, just by chance, happened to carry the T variant, right? That does allow you to digest lactose into adulthood. We don't know who they are; they're just there in the population. And that was the case until about 7,000 years ago, when we started domesticating cows, right? And so therefore, we had abundant milk in our societies, and this led to those individuals in green who had lactase persistence to have a huge advantage. This is how natural selection works, where suddenly individuals who could digest lactose found that it was a lot more advantageous for them, and this T variant was highly selected, and so gradually, in fact, rather quickly, the genetic makeup of the population changed. So from most people being lactose intolerant, a reasonable fraction of people then became lactose persistent in a fairly short span of time. And that's how we think lactose persistence evolved in humans, at least until 3 years ago. So suddenly, 3 years ago, there was a new study that changed our understanding a little bit. Researchers went and said, listen, let's look at archaeology. And that's kind of the cool thing about human genetics, because it ties in with other fields as well, right? Anthropology, archaeology, so linguistics, so on and so forth. So what these researchers did was they went and collected pottery from archaeological sites around the Middle East and Europe. And they found that in many of these pots, ancient pots essentially, you could actually see if there were milk deposits. There was very kind of small remnants of milk deposits existed in these pots. And they went and asked, okay, so in which of these regions was milk found? And they found that, a lot of regions around the world had these milk deposits. But in those same regions, the individuals who lived there did not actually have very high levels of the lactase persistence allele. And that didn't fit with the previous hypothesis, right? I mean, if milk apparently was common in these regions, but lactase persistence allele was not, is this selection hypothesis really, it doesn't even hold? And so it doesn't hold. So therefore, we have a new hypothesis right now, which is that lactase persistence may not have been super strongly selected when nothing else is going on. And it's something that a lot of us probably also experience. Many of us do not have the lactase persistence gene. Like in India, it's not super common. But I'm sure all of us, or most of us, actually consume a reasonable amount of milk. And that's because not having the lactase persistence phenotype in a time of plenty, in a time of normalcy, is not really that advantageous. Even being slightly lactose intolerant is fine. We can get by. But in times of famine, or if there's an epidemic or a plague, this mild diarrhea, the mild intolerance, could turn into something more severe. And it's in those periods that we think natural selection played a part. So natural selection did lead to that tea becoming really common, but it wasn't just because of domestication. It was because of domestication plus these periods of famine that happened, where the ability to digest lactose in adulthood became really, really advantageous. So this is our current hypothesis for why lactase persistence exists in humans, and this is actually one of the loci for which we have the clearest evidences of natural selection acting on human populations. I'll move on to the next chapter, which is similar, which talks about another genetic switch. And go ahead. Yeah. Hmm. To the end. Okay, so let's keep the questions to the end. I will come back to that. That's a very interesting question. And yes, I do want to get to that, so we'll come to that at the end. Yeah. Shall we come? I'll come to the questions at the end. You want to tell something? Okay. You read about this in the I Wonder magazine? Awesome. We will discuss I Wonder later. Yeah? Okay. Okay, so I do want to talk about another life-saving genetic switch, right? So one way of thinking about the previous story was that the switch in the genome, the presence of a C or T, can turn on or turn off the lactase enzyme. And there are many such switches, some of which can actually be life-saving. So beta thalassemia is a disorder, perhaps some of you have heard of it. It's when an individual has a mutation that doesn't produce hemoglobin, right? The protein that's required for blood cells to carry oxygen. And the lack of this adult hemoglobin leads to fewer blood cells, so you're tired. It's such an important feature of our bodies that the lack of this leads to a whole bunch of severe phenotypes, right? So you usually need regular transplants. The only real cure is a bone marrow transplant, which is very expensive and very painful. So how does this work? Let's again talk about this switching, right? So firstly, at the bottom, we actually have multiple hemoglobins. Even though we think about hemoglobin as one thing, we actually have what's called a fetal hemoglobin. So one particular protein that's only active in the very early stages of our lives. So the one in green, right? So it's produced by our cells when we are a few months old, and then completely drops. And what most of us have currently, the hemoglobin that we are producing right now, is adult hemoglobin, which is inactive early and then active later. How do our cells do this, right? So they do this using another switch, where how do our bodies know that at some point I want to turn off the fetal hemoglobin and I want to keep the adult one on? So there is another gene called BCL11A, the name doesn't matter, where this position in the genome, it acts as a switch for this gene, BCL1A, right? So if all of us have this region that tells our cells, listen, this person is beyond the fetal age, let's turn BCL11A on, which produces BCL11A protein, so that protein is in our cells. This protein now goes and binds to the DNA of the fetal, around the fetal hemoglobin, so it literally blocks it from being active, right? So this protein inhibits or stops fetal hemoglobin from being active, and it doesn't do that to adult hemoglobin. So scientists used this to come up with a very interesting idea. They said in individuals, if we don't have this adult hemoglobin functional, they have a mutation here, how about we try to keep this active? Instead of turning it off, just like with the lactase gene, right, which some people, it remains active in adulthood, can we keep this active in adulthood? And a study in 2007 showed that some humans actually have some variants in that region that keep that the BCL11A gene reduced. So naturally in the population, some of us have those variants that actually produce mild levels of fetal hemoglobin even in adulthood. So what researchers have done is then they used this knowledge to say, let's turn this off. Let's turn off that region in the genome. Let's delete it. And what that deletion will do is it will stop BCL11A from being produced, and in turn fetal hemoglobin will be produced, and an individual who cannot produce adult hemoglobin, an individual with a mutated form of the gene who would otherwise have thalassemia or sickle cell anemia, you now have fetal hemoglobin, right? So this is now an approved gene editing therapy. So gene editing is actually, I think it is equivalent to magic. The closest human beings have come to magic, because we can go into cells, change things, actually change the way our bodies function. And so because of sort of our knowledge of human genetics combined with our ability to actually perform gene editing in cells, we now actually have an approved cure for sickle cell anemia and thalassemia, right? So this just came out about 5 years ago. This is now our best strategy to cure these two disorders. The third chapter that I want to talk about is about mutations, because I think that's the immediate, we think about DNA, we think about gene, we think about mutations. So let's spend a little time talking about mutations. How do we get mutations, firstly? The most, the easiest answer to why we get mutations is when the DNA divides. Right? Just like when we are taking notes, someone's narrating something and we're taking notes, we make mistakes, we make typos. When the cell is dividing, our cells are constantly dividing as we are growing. Every time the cell divides, the DNA has to divide, and the cell makes mistakes. And so the DNA accumulates these typos, and those are what mutations are. We normally think of mutations as of two kinds. There's germline mutations, these are mutations that happen only in the sperm or the egg. If a mutation happens in the sperm or the egg, then that mutation gets passed on to the next generation of the embryo, and then gets passed on to the next generation of the family of the organism, right? So that's why it's called germline, because it happens in the germ cells. The other class of mutations are somatic mutations, where our DNA is also affected by environmental factors, right? I mean, that's why we don't want to be exposed to too many X-rays, etc. That's why we use sunscreen, because all of these lead to damages of the DNA. But these mutations usually affect a part of the organ that is that is mutated, but do not get passed on to the next generation. So these are called somatic. And what I'll talk about is a study that looked specifically at germline mutations and what they look like. So what this bunch of researchers just this year did was, this again went back to our favorite way of studying humans by looking at large families. And they collected the DNA of all of these families, sequenced all of these samples, and tried to look for how often does mutation happen. By comparing the DNA of a child with the DNA of the parents, what can we say? And what they found is that every individual has about 100 to 200 new mutations that the parents don't have. So I have about 100 to 200 mutations that you wouldn't see in either of my parents. So that is the rate of mutations per generation, right? And these are the kinds of mutations that lead to things like the lactase allele, the lactase variant arising in the population. So mutations are not a bad thing, contrary to what we think. If we try and think about which of these mutations are from my mom or my dad, we see that most of the mutations we carry are actually from our fathers. So about 70% is from paternal, from the father's line, and about 16 to 20% are from mom. And the remaining smaller percent comes in the first few stages after the embryo is formed. You know, the first few cells, mutations happen, and then they essentially are found in the entire individual. So this is the most interesting part of their study, I thought. So here, let's look at the green lines here. Here, what they try to see is, you know, we get told, have kids early, have kids early. So, and one of the reasons we get told have kids early is because, you know, we pass on mutations to our kids. How does that look? So if I look at mother's age and father's age, and look at how many mutations get passed on to the child, we look at the green lines, because these are the mutations that occur in the sperm or the egg. We see that father's age has a much larger effect on the number of mutations a child inherits compared to mother's age, right? So if a father is about 20 years old, the child on average gets somewhere between 30 and 40 mutations. If the father is 35, the child gets somewhere between 50 and 60. Whereas for mom, that line is a lot less steep, right? Mother's age matters a lot less than father's age, simply from the perspective of looking at the number of mutations that are passed on. And why is this? Why would the two be different? One would have expected that perhaps that both mom and dad should have an equal effect, right? And it's because of the way sperm and egg are processed very differently in the two sexes. So if you look at mom, so my egg cells were actually processed in my body while I was a fetus inside my mother's womb, right? So what happens is all of the eggs I will ever produce in my entire life were produced when I was a fetus and frozen at a semi-permanent stage. So essentially, my age does not add to the eggs' age because they're already produced when I was a fetus, right? So that's why maternal age has less of an effect on mutation rate, whereas for men, sperm are constantly being produced. So the older the father, the more the number of generations of the number of replications or divisions that sperm have undergone. And so therefore, father's age has a very direct relationship to the number of mutations in sperm. And that's why the older the father, there's a very steep increase in the number of mutations to children. I want to state one thing is that not all of these mutations are necessarily leading to disease, right? Genome, if you remember, is 3 billion letters long. These mutations we add about 100 to 200 per generation, right? So these 200 could be scattered anywhere in the genome. They may, very often, for most of us, they're probably not doing anything, or they are doing something we don't even know of. If you're really unlucky, some of these mutations could lead to a severe disease, but for most people, right, 100 out of 3 billion, most of them are really inconsequential. Yeah. How am I doing on time? I just want to make sure. Okay. So in the next chapter, I want to move beyond looking at families to looking at populations. Like what if I look at, I said it's becoming very easy to sequence everybody. So what if I just sequence 1,000 people? What would I see? What would that look like? We've been trying to do this for the last 2 decades in projects that are called 1,000 Genomes, where we sequence individuals from around the world and really try and understand what human genetic variation looks like. The first thing we learned is that, it's from projects like this that first fact came, which you've probably heard somewhere, right? Which is we are all 99.9% similar, which means that if I take my genome and somebody's genome in this room and compare all of the 3 billion bases, at 999 out of 1,000, they would be exactly the same. At 1 out of 1,000, we would probably have a difference, right? I might have a C, you might have a T, or vice versa. Another really interesting observation is that we are much, much, much less diverse than other species. So a bunch of researchers at Oxford found that if you look at the DNA of 2 chimpanzees from neighboring populations, and also look at genetic differences from 2 random humans from any parts of the world, the two chimpanzees are going to be much more genetically different. And this is because humans as a species are actually contain a lot less genetic diversity compared to most of the other species, most primate species. And this is because we started with a very small number, it's called a bottleneck, because of which our initial variation was so much lower that that has carried on to our species. This is something that's a very unintuitive thing, you know, I mean, we are so attuned to seeing differences in in faces, in skin color, in physical characteristics that we tend to amplify the differences in our head. But if you really go back and look at the biology, we're really a lot less diverse than we would think. And the last sort of similar point was that most human genetic diversity is contained within populations. And what that means is if, you know, a lot of people were assumed that your genetics would be able to easily separate what we call races, right? So let's say you would immediately be able to, you would have completely different genetics for an African population compared to an East Asian population compared to a European population. But what we now know is that the amount of variation that you see within a single population or a single continent is usually much larger than the amount of variation between continents. So this is, for those of you who are, who are statisticians, it means that most variation is actually found within a population as opposed to between populations. And this is again something really highlighting the fact that we tend to amplify differences based on cultural reasons, but biologically or genetically, a lot of that doesn't quite hold true. If I look at the number of variants in the genome, right? So if I look at all of our genomes and just count how many variants do you have versus I have versus somebody else, compared to some reference, okay? What do we see? What we see is that this group of populations in yellow, which are all African populations, tend to have a much larger number of variants than any other part of the world. And I don't know if this was, again, obvious to any of you, but there is a reason for this. A lot of what we see in our genomes can be inferred from our history. And the reason for this is what's called the out of Africa bottleneck. Humans arose in Africa, hundreds of thousands of years ago. All humans who are not African emerged from small populations in Africa. So essentially, small groups of humans left the continent and populated other parts of the world. And what that meant was that all of the diversity that was contained in the human population, out of that, it was only a small fraction that left and then populated the rest of the world. And this is called a bottleneck. And what this bottleneck means is that the genetic diversity is a lot less in every part of the world except Africa. So if you really study African populations, this is a huge wealth of genetic information that you would not capture if you studied any other population in the world. Another common area where we study these populations, right, is by looking at to study diseases, because I think everyone who thinks about genetics often thinks about diseases. So another very common experimental design is we collect a large group of people, some of whom have a disease, and we'll call them case, and some of them don't have the disease. We'll call them controls, sequence all of them, and try and see what are the differences. What parts of the genome are different in cases or controls? Or what parts of the genome perhaps have different numbers in cases and controls, right? And so the most common trait in which we do this is height, for the very biological reason that height is very easy to measure, right? And it's any study, the doctor will first ask you for your height, and so therefore you have millions of people around the world for which you have genome sequence data, and you also have height measured. So scientists took millions of people, put them together into one study and found, okay, so what parts of the genome are associated with height? Now, most of you have thought about genetics from a Mendel's perspective, where one gene leads to one variant, one phenotype, right? One gene leads to your P being round or wrinkled or green or yellow, but there's 12,000 genetic variants across the genome that influence height, right? So any of these 12,000 variants could either increase your height by a little bit or decrease your height by a little bit. And if you visualize that in a slightly different way, each of these dots is one of those 12,000 variants that influences height, okay? I've plotted here from very rare, so these are variants that are found in very few people. Maybe only one out of all of us in this room carry those variants. And towards the right are variants that are very common, right? So about half of us in the room carry those variants. And on the y-axis, or from top to bottom, or around the middle here, are variants that affect height, but to a very small amount, right? So maybe it increases your height by 0.1 or by 1 millimeter or by 5 millimeters, right? So it affects height, but by a very, very small amount, as opposed to maybe a variant like that top one on the right, which may have a much higher effect on height. So why do we see a plot like this? What we see is that variants that are found in many people tend to have smaller effects on things like diabetes or height. And this is because, going back to natural selection, right? If a variant was very advantageous, which means that it had a very high effect on something like height, it would be selected for, which would mean that everyone would suddenly have it or nobody would suddenly have it, right? But, and so therefore, variants that are common tend to have smaller effects on the phenotype. Whereas variants that are very rare, natural selection is not very good at acting on variants that are very rare. So if you have a variant that has a very, very high effect on the phenotype, chances are that it is very rare. It's not very commonly seen in the population. And so this is actually a pattern we see very often, which is that many of the variants from these disease-associated studies are actually have very small effects on whatever you're studying, like diabetes, let's say, or like cholesterol levels. And just to illustrate that a little better, what we wanted to see was something like this, right? Let's say a variant is affecting height. People with the variant, people without the variant. What we would love to see is they completely look very different. You can immediately predict a person's height just based on one genetic variant alone. But what we actually see is something like this, right? People who carry the variant, on average, they tend to have maybe be a little taller or be a little shorter, but they're really not that. These two groups are really not that different. So the effect of a single variant is usually not very high. So instead, what we're moving to now is looking, instead of a single variant, we're combining the effects of all variants. And we're saying, what if I calculate the combined risk from all of those 12,000 variants across the genome, and then use that to predict height? This is kind of the latest where the field is going in human genetics. It's called a combined genetic score or a polygenic score. And what this means is that for, as opposed to something like a severe disease like cystic fibrosis, what you would usually see is a person with cystic fibrosis would have, one variant in a gene that we know causes cystic fibrosis, whereas for something like heart attack or diabetes, we have many variants in the genome. This person has many risk variants across the genome, which collectively add to this person's risk for diabetes, right? And person 1 with heart disease may have this set of variants. Person 2 with heart disease may have a completely different set of variants. And the reason genetic scores are becoming more and are being studied more is, let's say I take all of our genomes again, I sequence everyone, and I calculate each of our genetic risk score, okay? And I plot it, and then later, after 40 years, I come back to each of you and say, okay, how many of you actually got heart attack, right? And what we see is that people who have a high genetic risk score have are 4 to 5 times as likely to get a heart attack as opposed to people who are in the lowest percentile of genetic risk score. So essentially, your genetic risk score, which can be measured at birth, your genetic risk doesn't change over your life, but your genetic risk can predict future onset of something like heart disease or diabetes quite well, at least for heart disease right now. So this is an area that people are excited about because this could lead to early intervention, right? If, for example, you knew that you had a high genetic risk score for heart disease, does that mean you would eat healthier? Does that mean you would maybe exercise a little bit more? Does that mean that you would be put on statins, which are a drug that people with high cholesterol take? So these are the things people are thinking about. It's not as successful for any other disease. So really, I showed you the example of the only disease, heart disease, for which this has been really successful. But right now, our efforts are to expand these to all other diseases and to see how successful these genetic risks can be. And essentially, can these be added to the clinic? So when your doctor asks you about family history, about all of your other risk factors, can they also look at your genetics? Finally, do I have time? Great. So I'll kind of move on to the last part, which so far I've kind of spoken about the successes of genetics and my favorite parts about the field. But I want to talk about potential challenges that any new field faces, and we seem to go round and round in circles as a society. So human genetics actually has a very gory past. The first human geneticists actually were eugenicists, who for them the goal was to use human genetics to, quote, uh, eradicate diseases, eradicate anything they considered bad from the human race. The Nazis were they really relied on genetic principles to say, let's get, quote unquote, get rid of people with Down syndrome, with schizophrenia, with things that, and really use genetics as the reason for a lot of things that are completely unacceptable. And all of this came because, because Mendel showed that in peas you have one gene that leads to one trait, and eugenicists and even all human geneticists extrapolated that to humans and said, okay, so one gene leads to this, one gene leads to this. Can we now use this to make society the way we want? So genetics has had to really move past the horrors of its own past over the last few decades. And we think we are doing okay, but now with our ability to sequence genomes and in IVF to sequence embryos, we have companies that aim to do something that are uncomfortably close to what I described. So we have companies like this which allow you to select your embryo, and of course, as parents, as potential parents, we'd want to make sure our kids don't have a severe disease, our kids are as equipped to face the real world as possible. But what if the choices that you're offered are things like intelligence? What if the choices that you're offered are things like eye color, height, right? This is, I want to make it clear, right? This is not a top-down approach where some authoritarian regime is killing people. This is a bottom-up kind of, it's a different paradigm where individuals are now have some information to make choices that do affect our species. I also want to point out that the science for this is not there. So apart from this being morally dubious, it's also scientifically complete rubbish. But the fact that these things are slowly becoming mainstream gives us things to think about. So I mean, I thought I would leave you not with answers about genetics, but more questions that I think many of us will have to think about as a society. So if we edit embryos, this changes the germline, and this leads to, essentially changing the genetic makeup of the human species. So who gets to decide which mutations are okay to be edited out and which not to? It's easier for some very severe diseases, where I think a lot of us would agree that for the most severe diseases, we would not want somebody to suffer. But what about something, again, like Down syndrome? In many countries with embryo screening, Down syndrome the frequency of individuals with Down syndrome has come down. But individuals with Down syndrome in these societies have said, the fact that the governments or societies are encouraging these embryos to not be selected makes us feel unwelcome. So should societies be more inclusive towards people with disorders, or should we preemptively be very conservative in embryo selection or gene editing? I think that's a question for society to think about. From a more practical perspective, for all of us, if you've had your genome sequenced, how do you control who has access to your data? I started out by talking about how genetics is sort of the most personal part of you. If I know your genome, I can, there's a one-to-one correlation, right? Your genome uniquely identifies you and vice versa. So would you be comfortable with it being accessible by researchers? Or what about if you haven't gotten yourself sequenced because you're a privacy junkie, but your sibling loves technology, gets themselves sequenced, puts their DNA up on a database that is public? Now, essentially, that means your DNA is available, right? Because 50% of your genetic information has been released. So how does one control that when your privacy is not in your hands alone? I mean, that's how companies like 23andMe tell me that my fourth cousin, they've identified a fourth cousin or a third cousin, right? Because, because we share so much DNA. Where does another prickly question is, where does insurance come in, right? If your information is available to insurance companies who know you have a high risk for Alzheimer's or a high risk for, let's say, early heart attack, what would they do with this information? And should they be even allowed to use this information? Right now, I think that India was fairly early in enacting policies for the last one, saying they do not have access to this information, but these are ongoing questions that are subject to change. And I think these are questions that we should constantly be thinking about, because as gene editing becomes becomes easier, and as we continue reading more and more more genomes, these questions are really going to come to the forefront, and they're not going to go away anytime soon. So I'll stop here and be happy to take any questions. Thank you.